Great, thanks. So this topic is an extension of spatial capture-recapture modeling that accommodates a really common data situation, and this work is in collaboration with Angela and Andy. So one of the common ways that wildlife biologists collect animal encounter data on a landscape is through non-invasive genetic sampling. So here we have hare traps distributed across western New York. We're trying to capture a fisher. And the problem with these data is that the hair samples that result are a poor quality DNA source. So in our example, about 50% of our samples are able to yield DNA that we can then use to genotype and create an identity for that capture. So typically, spatial capture-recapture models only use the samples where you have an identity. These are these red dots here. The yellow dots are an unknown ID capture where we know it was a fisher, but we don't have the individual. Those data are typically thrown out. Well, we wanted to see if this information could actually add value to our modeling, whether it's understanding landscape variation in density or improving population estimates. So what we do is we simply assign an identity to each unknown sample probabilistically based on the location. So here we've got two traps with unknown captures, and the probability that that capture is assigned to a certain individual is based completely in space. So obviously this trap on the left here is probably the red individual, the trap on the right is probably the blue individual. And what we find is that indeed if you include these unknown captures, you can gain some precision in the population estimates, but what we need to do is understand better how these models perform through simulation, and that's really why I'm here today, because I use the Atlas cluster to run a bunch of simulations, and it takes a whole lot of computation to do that. So come see my poster if you want to talk more.